So today our group would like to present the evolution of Xylanase in enhancing the breakdown of untreated oil palm trunk. So let me introduce my members, Atikah and also Fadilah.
Okay, thank you, Sun. So before we proceed further in this presentation, do you know what is the characteristic of the enzyme? So let me explain. First, enzyme is a biological catalyst because they can speed up the reaction. Second, enzyme is extremely a complex molecule because when enzyme bind to its substrate, it will form enzyme substrate complex. Thirdly, enzyme is linked together in a long chain and fold up into 3D structure because it is determined by the sequence of amino acids that held together by the hydrogen bonds. Next, enzyme is built up for the 20 different kinds of amino acids that held together by peptide bond. Lastly, enzyme has a specific binding site, so only a specific substrate will fit to its active site. So here I have a two theory of enzyme action involved. Firstly is lock and key hypothesis. Second is induced fit model.
Thank you, Fadilah. So now I will proceed with the next point, which is how to improve the efficiency of enzyme by evolution. Before I explain further about this point, let me tell you what is the general meaning of evolution. Actually, evolution is a development of any kinds of living organism from a phase to another next phase. Back to the point given, actually the main purpose we do evolution is to improve the efficiency of enzyme, right? So, we use Xylanase as the main enzyme in this evolution. To proceed, we need to use DMF as to create mutation in enzyme genetic codes. Then introduce it to the bacteria that produce thousands of different variants of Xylanase. Now the challenge is to find out which of these variants work better in DMF or in evolution. We call it a selection. We know that Xylanase can break down into xylan. So, we take the variance of Xylanase and put it into 35% of DMF. For the next step, we introduce a new round of random mutation which yield them to work better in DMF. In third generation, we can find a variant of Xylanase that works 256 times better in DMF. So, it means there are combination of 10 different mutations.
Thank you to Atikah. And now, is it possible for us to create desirable enzyme? Yes, it is. How? By mating the gene in a test. First, we introduce random mutation to the gene of enzyme that we want to change. Then, the mutated gene of enzyme will inserted into the bacterial cell to let the bacteria replicate the gene. After the gene has been expressed into the protein which is the enzyme that we want, the enzyme will be tested. The most efficient at catalyzing desired chemical reaction will be selected. To gain more effective enzyme, new random mutation will be introduced. This cycle will begin. The method that is commonly being used to make mutation is DNA shuffling, which is a method to rapidly propagate beneficial mutation in a directed evolution experiment. It involves the digestion of several genes into random fragments. Then, with the help of tools of DNA technology, the random fragment will be puzzled together into a complete recombinant gene. Using several cycles of DNA shuffling, we changed the enzyme so that it became much more effective than the original one. This recombinant can result in the even more efficient enzyme.
So now I'm going to talk about the new enzyme, how to increase and contribute to the efficiency of degrading untreated oil palm trunk. So the first one is it can reduce the production of untreated waste product. For our example is the oil palm trunk. So the second one is the developed Xylanase can maximize the production of reducing sugars such as xylose and glucose, which is much more important in the industry. So the last one is it can produce highly efficient Xylanase in reducing the waste product. So in conclusion, the new enzyme can help a lot in our daily life. So thank you.